Hi students, how are you? Yes, now we are going studying about food preservation. We have already learned that affections of poisonous substance of food poisoning, the harmful microorganisms destroy food particles, etc. Then we already know that, some briefly know that about food preservation. Okay, food preservation is very important of. Then in food preservation, what is the importance of food preparation? It is protecting from microorganisms, uh, some like bacteria, virus, fungus, and rats, some reptiles, lizards, uh, moisture. Okay, so food were prepared and storing without any damage. So we will discuss about the food preservation. Yes, there are five methods of food preparation methods. First one is chemical methods. Yes, first is chemical methods. Food preserved by using some chemicals. That chemical is known as preservatives. Preservatives. Yes, have you noticed about preservatives? Food materials free from microorganisms like bacteria, fungus, some rats. Okay, they using some chemical substance, that is preservatives. Okay, then the second one is by common salt. This is a traditional method one. Traditionally, we are using salt for preservative. Okay, fish, dry fish, some fruits, vegetables, etc. We are preserved by use of common salt. And then by sugar, we are using sugar for preserving some food materials like jams, jellies, cherries, fruits items, etc. We are using sugar. And then forward by oil and vinegar. We know about vinegar. We are using vinegar is acetic acid. Vinegar is acetic acid, diluted acetic acid. We are using vinegar for what? For food making, some like meat, like food productions is mainly using vinegar. And oil and vinegar using some materials, some food materials like lemon, ginger, mango. Yes, these are preserved by oil and vinegar in the form of pickles. In the form of pickles. And some fish meat, then husbandry, animal husbandry meats, poultry meats. These are preserved by method of oil and vinegar, just like pickles. The last one is heat and cold. Example, milk. Milk is heating. So, the turning into the curd, that process is will reduced because bacterial actions, bacterial process, that will reduced when heating the milk and cold, and then milk is cooling because the bacteria, the life structure of bacteria, the living temperature of bacteria is around 28 degrees Celsius to 38 degrees Celsius. So when we cooling, that means it been below 28 degrees Celsius. The bacteria and larvae will destroy, okay, will inactive, and we heat, then bacteria will destroy. So the food preservations are in 5 types. First one is the chemical methods, and second is the common salt, and third one is by sugar, and fourth one is by oil and vinegar and heat and cold. This is the food preservation. Next is the last part of the chapter, that is nitrogen fixation. Nitrogen fixation. Yes, we know about nitrogen. Around 78% of nitrogen within the atmosphere. The atmosphere is filled with nitrogen about 78%. 21% of oxygen here. 21% oxygen, and remaining are carbon dioxide, some other dust particles, etc. Okay, then this atmospheric nitrogen, the 78% of atmospheric nitrogen cannot absorb directly by the plants or animals, but nitrogen is very essential element for plants for growing. Nitrogen is very essential element, but the plants have no capacity, no ability to absorbing direct atmosphere from atmosphere. Okay, then how is it possible? There are 3 methods are here. The conversion of nitrogen. The first is when during lightning and thunder. The first method is lightning and thunder. Lightning and thunder. The rainy season, when occurs lightning and thunder, the atmospheric nitrogen will divide into nitrate. Then that atmospheric nitrogen is dissolved into rainwater and falling in the soil. So the atmospheric nitrogen is converted into nitrate. Soil is an atmosphere. Okay, during thunder and lightning, atmospheric nitrogen dissolving rainwater and going through its soil is atmospheric nitrogen is converted into nitrate, that is NO2. This is the process of nitrogen fixing. This is process of nitrogen fixing. Nitrogen fixing. This is the one method. And the second method The second method of nitrogen fixing is some of leguminous plants have root nodules. Just like look that this is stem and this is root. Root have a root nodules in it. In these root nodules, there have a position of some bacteria, that is useful bacteria. That bacteria's name is rhizobium. Name is rhizobium. These bacteria can absorbing atmospheric nitrogen and convert it into nitrate to soil. Okay, look, some of the leguminous plants like pea, black gram, etc. There, the root have root nodules. That is the living place of some bacteria. The bacteria is rhizobium acetobacter. Okay, this rhizobium bacteria can absorbing atmospheric nitrogen directly and is converted into nitrate. The second method is by the using the working of legume plant. Leguminous plant with the help of bacteria is called rhizobium. So this rhizobium bacteria is otherwise known as nitrogen fixing bacteria. This bacteria is absorbing atmospheric nitrogen to soil nitrate. This second method of nitrogen fixation. The first one is lightning and thunder, and second is leguminous plant. And third one is some algas. Some algas that were aquatic organisms. Some organisms like aquatic organisms like algae, it can be converted nitrogen to nitrate. It can be absorbing atmospheric nitrogen to nitrate. Some algas, it can directly absorbing atmospheric nitrogen to converting to nitrate. This is not in the soil. This is in soil. This is in soil. But this is in aquatic medium. That means water. There's three types of nitrogen fixings here. Yes. First one is the lightning and thunder. Atmospheric nitrogen directly dissolving the rainwater and then reach the soil nitrate. Then this nitrate can be absorbed by plants for growing. Okay. Then the second is leguminous plant. With the help of leguminous plant like pea plants. Yes, it have root nodules. The root nodules is the presence of bacteria. The bacteria's name is rhizobium. Rhizobium is nitrogen fixing bacteria. This bacteria, rhizobium acetobacter bacteria, it can converting atmospheric nitrogen to soil nitrate. And the third one is alga. Alga is in the aquatic plant. It can absorb atmospheric nitrogen to water, aquatic nitrate. This is using the aquatic plant for growing. Okay, this is the nitrogen fixation. Look. Yes. Next is nitrogen cycle. Cycle means every cycle is starting with one point and ending with same point. That is cycle. This is the nitrogen cycle. Yes, nitrogen cycle. Nitrogen cycle first step means nitrogen fixation. Nitrogen fixation means two type of nitrogen fixations are here. We have already learned nitrogen fixation. The first one is lightning and thunder, and the second one is the bacterial action, that is bacteria is rhizobium. Then atmospheric nitrogen is converted into soil nitrate. This is the first nitrogen fixation, first stage, that is nitrogen fixation. Then in this time, nitrogen in the soil in the form of nitrate. The nitrate is uptaken by plants. Nitrogen is uptaken by plants for growing, for plants activities. Okay. Then this nitrate now the position of nitrate is in plants. Then the animals eat this type of plants. Animals eat these plants. Then nitrate is in this animal. Then these animals and plants are dead or decay. In animals, like nitrogen is based from excretion and death. Plants or animals have formed nitrogen based and from excretion and death. Then this nitrogen will comes to again soil. Within plant and animals, the nitrogen will comes to soil. And then now the nitrate is in return soil. Okay? Then the return nitrate, this nitrate is acting with bacteria. That bacteria is denitrifying bacteria. Okay, it's not it denitrifying bacteria. Nitrifying bacteria. Denitrifying bacteria. That is, soil nitrate is converted into atmospheric nitrogen. This is the nitrogen cycle. Look again. Atmospheric nitrogen is in soil nitrate converted by method of nitrogen fixing. The two type of nitrogen fixation. The two type of nitrogen fixations are during lightning and thunder, and the bacteria like nitrogen fixing bacteria, that is rhizobium. With the help of rhizobium in leguminous plants, helping nitrogen fixation. That means atmospheric nitrogen into the soil nitrogen. Atmospheric nitrogen converted into the soil nitrate. Okay, this is the first step. And the second step is soil nitrate. Soil nitrate is uptaken by plants. Soil nitrate is uptaken by plants, and these plants eat by animals, and then animals excrete nitrogenous waste and they wear death and decay. Always plants excrete some nitrogen waste when plants dead and decay. That time this nitrate will return to soil, and then then atmosphere nitrogen is in the soil nitrogen, and finally this soil nitrogen is converted to atmospheric nitrogen with the help of denitrified bacteria. What is the importance of denitrified bacteria? The denitrifying bacteria is converted soil nitrate to atmospheric nitrogen. This is the nitrogen cycle. Okay. Yes. Then revise this chapter again. And but for you just keep a biology notebook must and write down the main points and questions and answer, and keep your doubts and asking with me, and then learning very well. Okay, then the chapter is now again revised. Look, the main points are in the board. The chapter name is Microorganisms Front and Foe. Foe means enemy. That's the meaning of foe, enemy. Okay. This microorganisms they are the smallest organisms. They cannot see the direct contact of eyes. Some of the most important of microorganisms are bacteria, fungus, protozoa, algae, etc. Protozoa, algae, etc. Bacteria. We know about bacteria. Bacteria are various types. E. coli bacteria, spindle bacteria. Sometimes bacteria are here. In there. And then the second is the fungus. Fungus. You know about fungus? Yes, we are seeing daily, regular ways. In a daily life, we can see fungus. From where? Yes, some bread mold is smallest blackish grayish color in the top of the bread. There is a powdery substance, that is fungus. Then protozoans mainly in aquatic organisms. Amoeba, paramecium. These are the examples for protozoans. Then algae. Algae are some photosynthetic organisms. They are not directly interrupted animals. And last is virus. Virus is very dangerous microorganisms because they can live and they can live any organisms like bacteria, algae, everywhere they can live. But the main thing is the virus is deactive. That means not living. Is look like a dust particles in the atmosphere. When the virus came into a cell, they can act. So virus is very dangerous one. Have you heard about some names of virus? Please note your notebook. Okay. Then in this chapter, mainly this microorganisms classified as two: friendly microorganisms and friend and foe. That is chapter name: friendly microorganisms, and foe means harmful microorganisms. Friendly microorganisms, that's the first part. Friendly microorganisms. These are using commercial uses of microorganisms. Commercially using microorganisms for what purpose? For beverages. That means production of alcohol. That way, with the help of these microorganisms, bacteria. And next is using medicines. Medicines. Some type of antibiotics are prepared from microorganisms. Some bacteria are using antibiotics. And then third one is vaccine. Dr. Edward Jenner discovered the vaccines of some disease-causing organisms from some fungus, from other microorganisms, especially poliomyelitis. It is a very dangerous disease. But now we can take vaccines regularly and we destroying and removing the poliomyelitis disease in the world. But nowadays the vaccine is very important one. The vaccine is made with microorganisms. And next is soil fertility. That helps soil fertility. We have already know about soil fertility, increasing the soil fertility. So how? We just learned in this class when the soil fertility is possible by the using of nitrogen fixing bacteria like Rhizobium, Acetobacter, etc. And then some cleaning the environment, some plant parts, animals, dead bodies, etc. They were decaying and decomposing with the help of some bacteria. This bacteria helping clean us our environment. This is the main friendly microorganisms. And social fertility, we have already know about vermicomposts, etc. We are using earthworms, some organisms decomposing with the help of some bacteria. They increase soil fertility. And look, these are the friendly microorganisms, and we are using some microorganisms in our householding, that's making idli, dosha. They will, you just looking that way. The flower will be puffs in size because of the presence of respiration, anaerobic respiration. Anaerobic respiration is done with microorganisms like bacteria. So carbon dioxide is filled with this flower. So that is in dump shape. Okay, this is, and milk is turned into curd. These other examples for friendly microorganisms. Sometimes we are able to create some microorganisms activity. So example is milk turned to curd. And next is the harmful microorganisms. Many of the microorganisms are harmful to human body, harmful to other plants and other animals, etc. That is a disease causing. Simply words, these are pathogens. Pathogens, the disease causing organisms. The disease causing microorganisms is commonly known as pathogens. The study of pathogens is called pathology. The study of pathogens is called pathology. Yes, this disease causing to animals and plants. Now about bacterial disease, fungal disease, protozoan disease, viral disease. Yes, bacterial diseases are there, and examples for protozoal diseases. And please write your notebook. We already learned the disease causing microorganisms and how we spread that disease. Yes, The last is food poison. Food poison. Yes. Some food is acting with microorganisms. They will be poison substance. They produce some toxic substance, then the food will be poisonous. Okay, the poisonous substance eating it cause to death. Okay, this is the main points of microorganisms and front and fore. That is the main points of this microorganisms front and fore. Okay. Okay, students, you must keep it your notebook already, I said, and let us be concluding this chapter, microorganisms front and fore. Okay, see you tomorrow. Keep well, learning well. Okay, bye.